got the study guide here. It's all marked up too. Alright? So all of these pink marks here indicate that I've generated a test question already.
Can I borrow that for a second?
No. Well, there's no notes. I'm just ticking off my points here. Alright? So,
It's all pink.
alright, any questions on chapter 6? Chapter 6 is dealing with movement across that plasma membrane. Alright, so you gotta be able to know passive versus active, what requires energy, what doesn't require energy, what requires a protein channel, what doesn't. Is it going from a high concentration to a low concentration, or from a low concentration to a high concentration?
So on the various types, do you just want us to know, like,
Things like osmosis, simple diffusion,
okay.
facilitated, active transport.
Okay.
All right, examples are going to be important, okay? Certainly know those cell-to-cell connections, so those gap junctions, adhesive junctions, tight junctions. I like tight junctions. I like tight junctions.
Say it one more time.
I like tight junctions. Okay. All right. You know, what's the difference between endo and exocytosis?
Are you asking us?
Yeah, that wasn't rhetorical.
Oh, okay.
Inside the cell, one's outside
Endo's
the cell.
inside, exo's outside.
Be a little bit more specific. Okay, yes, so things are being secreted out to the cell is exo, things that are being brought in is endo, alright? You know, we talked about resting membrane potential a lot in both 6, I introduced it in 6, and then we talked a lot more in 7. So really understand why that value is negative. Alright, understand that concept.
Can you repeat that question, please?
So understand why you have a negative resting membrane potential.
No, it's because of the protein.
Why do you?
Because of those proteins.
Okay. Yes. Okay, so what about those proteins? And where are they found?
Inside the cell.
Okay. Now, what are your other 2 players?
Sodium and potassium.
Potassium and sodium. Are they both, what's their charge?
Positive.
And
equally distributed.
How?
More sodium goes out of the cell and less potassium goes in.
But how?
3 sodium for 2 potassium.
What is causing that unequal return of ions?
That's the pH
Very good.
Oh, that's simple,
Yeah.
okay, I thought it was like a
And
more
that's-
in-depth answer than that.
and that's 3 sodium coming
for 2 potassium.
going
Yeah.
out and 2 potassium coming in, right?
Mm-hmm.
Are you asking me, or are you just trying to reiterate
Both.
and being hesitant about it?
Both.
Okay. Alright. So, here's my question, and you know this answer already because I ask it so many times. Without the sodium potassium pump, alright, what is the direction of those ions? So sodium going in, why? Concentration difference, so sodium is highest where?
Outside.
Okay, potassium,
It's highest inside, so it'll go out.
and it goes out. And so that sodium potassium ATPase pump returns those ions from where they originally came from, but it just happens to do so unevenly. Alright, you got that better than the 11:30 class. I about had a heart attack there. Alright. Chapter 7, neurons and synapses. What are the 3 parts of the neuron?
Cell body, dendrite, axon.
Okay, so what do each of those things do?
Dendrite receives the information, cell body translates it, and then axon transports it.
Okay. Alright. What are those 2 types of transport along the axon?
Head. So,
Nope.
Active transport.
Nope. Nope.
What was
Are you
the
talking about like
question
the
again?
fast component, slow component?
EPSPI, APSP.
Well, that's part of it, but there's 2 major
Anterograde
anterograde
and retrograde.
okay, so what does that mean?
Integrated
Your
cell to
big
axon
cell
and
and
dendrites, and then retrograde is axon and dendrites toward the cell body.
Okay, alright. Tell me something about a bipolar neuron. You just saw one 5 minutes ago.
One dendrite, one axon.
Alright. Alright, name 2 neuroglial cells of, well, I shouldn't have said that. Name the neuroglial cells of the peripheral nervous system.
Schwann and so.
Okay.
Satellite.
Schwann cells do what?
Form the
Myelin sheath.
on the axon.
And the satellite cells?
Support the
Support neuron cell bodies.
Okay. How about those of the central nervous system?
Microglia.
Okay, what's microglia?
It's like.
That's the immune system.
Immune system.
Immune cells?
And then the oligodendrocyte is the myelinated sheath.
Okay, what else?
That was called
Astrocytes.
oligodendrocyte.
Ependymal cells do what?
They line the ventricles.
Epithelial
and
cells.
Myelin. I don't know.
No, definitely not myelin.
I don't know.
Epithelial.
It's some
They produce a fluid.
CNS?
CNS.
CSF.
Oh,
CSF.
Yeah, cerebral spinal fluid. Okay, what's the last one I heard it?
Astrocytes.
Astrocytes. What do they like to do?
Regulate extra.
They regulate a bunch of things to help those neurons
Yeah.
stay
Primary,
healthy.
what is the primary what? I'll take the
There's
primary
the
something.
most abundant.
No, the most abundant.
They're most abundant.
In
Very
the
good.
cell in the CNS.
All right. So tell me something. Help me draw an action potential. Okay, I'll draw the action potential. All right. So, what about it? Why does it go up? Why does it go down? Why does it go further down and then peter out?
Depolarization.
Where's that at?
What's going up?
Going up.
Okay. So during depolarization, what is happening?
Sodium goes into the cell.
How?
Sodium channels open.
The gated
Voltage
channels.
channels.
Okay. Voltage gated sodium channels do what?
Open.
Open during depolarization. All right. When do they close?
During repolarization.
Where on the graph?
Down.
Down.
Right.
Apex.
At the top.
Did you give us that lab back that has that?
I sure did.
Did you?
So voltage gated sodium channels close. Okay? Alright, that right there is the end of depolarization. What happens next?
Repolarization.
Here?
Yeah.
Okay, so what happens there?
Voltage gate potassium channels.
Voltage gated potassium channels open.
It's a mouthful.
Alright, so there's no sodium here. Sodium is done. Sodium was finished at the top where that big blue dot is. Okay? Now, what happens after repolarization?
Hyperpolarization.
Where?
At the bottom. It dips back
The
up.
dip.
The dip? Okay. Alright? So when do those voltage gated potassium channels close?
Relative.
Bottom.
Way at the end.
Alright, so it's going back up.
I just saw that on here.
No.
So.
Okay? Why? Okay, so here's your bonus question. Well, actually, not getting a bonus. Why would you exhibit hyperpolarization?
Because of the sodium.
And
The harder to
they
close.
close slower. Potassium channels are slow. Alright? So that brings me to the refractory period. What are the two types?
Relative.
Absolute.
Absolute.
Okay, so absolute is what?
Absolutely nothing can get it back up.
Right.
No action potential. I don't care if it's like the power of a sledgehammer. There's no action potential. Alright, so where would you find that on the graph?
It's right before your
Roughly.
voltage gated.
Okay, so right about here. Okay? This is absolute. Okay? It's absolute because during this phase, those sodium channels open and close. and they don't like to be re-stimulated, okay? What's the next one?
Relative.
Alright, so obviously that's over here. So relative means what?
It's still recovering, but I can get
It's-
action potential with a great stimulus.
Okay, it's still recovering, because those voltage gated potassium channels close slower, but as Tessa said, you need to have a strong stimulus, stronger stimulus, in order to get an action potential.
So absolute, refractory period is like straight down on the down slopes.
I mean, it encompasses a little bit of the downslope, but not completely. Really, absolute is going to deal with the fact that you can't stimulate those sodium channels to open again.
Do we need to know this graph, or this, whatever this example for the test? Like, is something like this gonna be on the test, or
We label
it's
each
like
part.
an actual picture of this gonna be on the test?
Or
If
just
it's
need the
not-
words.
If
Labeling
not,
is too easy
you
at
don't
this
have to,
point.
you don't have to add one or anything, I'm just asking.
I'll ask you questions about that action potential, for sure.
Just like what we went over.
Can you just tell me what numbers 1 through 50 are? That'd be much easier.
Flip it around again.
Now,
You see that real quick.
alright. So understand, and it seems like you do, so just make sure you don't forget it by Monday. Alright? Now, if I'm an action potential and I've reached the axon terminal, alright, what's at that axon terminal, first of all?
The,
Synapse.
Synapse. Alright, so what would be on the axon side? Synapse
Right.
connects the neuron to another cell, does it not?
Synaptic cleft.
So the signal, okay, so the signal from the presynaptic has to make it to the postsynaptic. How?
By a cleft.
Neurotransmitters. So you're gonna find those synaptic vesicles loaded with neurotransmitters at that axon terminal. So if I'm this action potential and I've reached the end of that axon, what's the first thing that's gonna happen? More specifically, what kind of what do you mean calcium gated? Voltage gated, alright? Voltage gated calcium channels do what?
and then calcium comes in.
Okay, open and calcium comes inside, so calcium does what?
Pairs with, or bonds with the G protein, binds with G protein.
Fuses with the cell membrane.
None of these are what you're looking for.
Dang it.
It's gonna stimulate it, so you can tell me that it binds with some sort of sensor protein, but together, what are you stimulating?
The synaptic cleft.
Okay, you need to bridge your information now, okay? So you've got these synaptic vesicles, how are they going to get rid of that neurotransmitter?
They find some.
Well,
If
Excited.
it binds to what? What will it
The
bind?
border and
What's the border of a cell?
cell membrane.
Cell membrane, plasma membrane. And so then the neurotransmitters are released by what process? Exocytosis, and they go to the,
Synaptic cleft.
alright.
I know that one. I participated.
Okay, you participated, very good. So the synaptic cleft, alright, here you've got these neurotransmitters moving along, and so they end up on the postsynaptic cell or membrane, alright? And so for a signal, the action potential to be carried on, what will those neurotransmitters do? Generally speaking,
Bind to a receptor. on the
bind to a receptor and hopefully cause
an
an action potential to occur. Alright?
We're so smart.
You are. You know it. You know it. Okay? I'm just
Yeah. Yeah.
bombarding you with questions and making sure you know it. Alright. So kind of tell me the difference between an EPSP and an IPSP.
EPSP is
increases.
excitatory. IPSP decreases the chance of an action potential.
EPSP increases.
Okay.
The
how?
Sodium and calcium
Okay,
Helps with
and
the
IPSP
decreases.
decreases what?
Action potential.
The chances of an action
Yes.
potential to occur by doing what?
Secreting chlorine.
Cl.
Chloride.
Chloride?
Chloride.
So is chloride coming in or coming out?
And
Okay, so it causes what?
Hyperpolarization.
Hyperpolarization. Alright, you're going farther and farther away from threshold. Alright. Okay, so what are the 3 ways of synaptic integration?
acetylcholine.
That's just a neurotransmitter.
Dang it.
The 3 ways that you can undergo synaptic integration. Convergence, so let's revisit that. What does that mean?
Divergence.
That's where two,
Doesn't have to be 2.
or multiple,
From double membrane.
neurons meet one.
Meet and synapse on one, okay.
Divergent.
Divergence.
Inflammation.
Give me some.
Okay, divergence, okay, I'm trying to,
Wait, let's finish convergence. So
Okay.
that was convergence.
yes,
Are we moving on to divergence?
yes, so what's divergence?
Divergent is whenever it's going from 1 to
One.
2.
Neuron.
Or,
1 neuron to 2 neurons.
okay, so that information parcels out into various neurons.
Yeah.
Alright, summation,
More than one stimuli causes the action potential.
okay, what kind of summation do you have?
Temporal and spatial.
Alright,
Oh.
boom, okay. Okay, chapter 7, done.
Yay.
10 minutes to go. Alright,
It's spatial, right? Temporal and spatial.
Yeah.
tell me something about the reflex arc. What are those components? In the right order.
Sensory receptors.
Interneuron, motor neuron,
And motor receptors.
And what?
effector organ.
An effector organ. Okay.
Can you say this? Can you say that again?
Oh, yes.
Just the order, but do you need to know anything else about it?
Well, you kind of already know a lot of things about it.
So it's
Can you say that order again?
Sensory receptor,
sensory neuron,
sensory neuron,
motor neuron, motor neuron, and then effector organ.
Effector order.
Okay.
Okay.
so now tell me this.
Is that chapter?
Which part is going to encompass the afferent side? AFF.
The sensory.
Sensory neuron.
So the sensory neuron, how about the efferent?
The motor.
I don't know.
Alright. La-di-da. What is the relay center of the brain called?
Thalamus.
Thalamus. Where would you find it?
Center brain.
Diencephalon. Alright. You know that the limbic system is going to be important for memory and emotions. Alright. Alright, here we go. Those autonomic neurons again. Alright. Quiz question. All preganglionic neurons, acetylcholine, is it excitatory or inhibitory?
Either or.
Well, if it's preganglionic,
It's
Excitatory.
it will be excitatory. If it's inhibitory, then there's no point in having an autonomic nervous system. Alright. Now, if I am a preganglionic neuron of the sympathetic nervous system, where will I synapse?
Terminal.
Eh,
Target tissue.
eh,
Target organ.
eh. Preganglionic sympathetic neuron will synapse in one of two places.
Sympathetic chain.
The sympathetic chain and,
You know, the other one.
The autonomic ganglion.
well, they're all autonomic ganglions.
I can't remember the name.
Collateral. The collateral ganglion.
Yeah.
So what's the difference between that again?
So the sympathetic chain is going to be immediately after the spinal cord. So on either side of the spinal cord, you're going to have sympathetic chain ganglia. So these preganglionic sympathetic neurons are coming off of the spinal cord and will then synapse either in the sympathetic chain or they go through it and synapse at the collateral ganglion, where you find it roughly in the pelvic and lower abdominal region.
And that's just in the sympathetic.
Sympathetic nervous system.
Yeah, there you go.
All right. Preganglionic parasympathetic neurons will synapse where? The terminal ganglia, which is very close to the effector organ. So preganglionic parasympathetic neurons are very long. All right. Okay, so if I'm a postganglionic sympathetic neuron, I'm going to secrete what kind of neurotransmitter?
Epinephrine and norepinephrine.
Okay, epinephrine and norepinephrine. Is it going to be, what kind of receptor do these neurotransmitters bind to?
Nicotinic. Wait, I might have jumped the gun there.
You did.
Messenger.
I'm sorry, you did.
Mesotonic. Make it up right there.
Okay, well let's
You're
save Isaac here. A nicotinic receptor is going to respond to what kind of neurotransmitter?
Acetylcholine.
Acetylcholine.
Adrenergic.
Adrenergic. Okay.
3 times. Thank you.
Sorry. You need to speak up. All right. My auditory cortex
Okay.
is like. All right. Okay, so let's go back. Let's talk about acetylcholine receptors or cholinergic. All right, the two types. One is called
Nicotinic.
nicotinic,
Muscarinic.
muscarinic. Now, is nicotinic going to be stimulatory, inhibitory, or both?
Excitatory.
Excitatory,
Always.
always. Okay. Muscarinic,
Both.
both, depending on the organ
Yeah.
in question. So, how can a muscarinic be excitatory? What will it do? How do you excite anything?
By releasing something.
How?
Release.
Sodium. All right. What about its inhibitory action,
Releasing chloride.
but more of
The postganglionic side.
closing potassium? Closing potassium hyperpolarizes. Okay.
So when you asked what norepinephrine and epinephrine, postganglionic, whatever two, and I said nicotinic, I was wrong. What was the answer to that?
What is the answer?
Adrenergic?
Adrenergic.
I don't know how to say that.
Adrenaline. That's another way of saying epinephrine. Adrenergic. All right.
And what's the other adrenergic?
I'm sorry.
There's another adrenergic one. What's that
My
one?
notes are very all over the place.
There's adrenergic
Oh,
and
yeah,
cholinergic.
Okay.
Right. All right. All right. Few minutes left. Tell me what a generator potential is.
We still on the same chapter.
No.
We're at 10.
10. The generator potential. Is it the same thing as an action potential?
No.
No. Can it cause an action potential?
Yes.
Only if it reaches threshold. I'm sorry, what did you say? Okay. All right. Can you tell me tell me, I'm not gonna say can you, tell me the difference between a tonic versus a phasic receptor.
Tonic is adapts to the stimuli.
In what way?
Slowly.
Slowly?
Slowly.
Okay.
Okay, phasic.
Adapts to it quickly.
All right, so temperature in general would be a good example of phasic. So if this was really scalding hot coffee, I might say, ooh, it's hot. Then after a while, that kind of heat sensation dissipates because those receptors have adapted, right? Pain receptors tend to be more tonic, right? Anything else? Oh, the type of sensory receptors for taste and smell. What are they called?
Gestation. Respiratory.
Well, not this type of cell, but the type of receptor.
Chemoreceptor.
Chemoreceptor. Okay. How about for hearing and equilibrium?
Oh, crap.
Mechanoreceptor.
Mechanoreceptor. Very good. Alright. So how do you feel about that?
Better than what I thought.
Well, it's new, alright?